Indeed, it is a great pleasure to be here in the Department of Biochemistry, Davangere University. I thank Professor and Chairman, Department of Biochemistry, Kumar sir, having invited me here to give a talk and in this special lecture series. And also I am very much pleasure to share the dais along with the Professor K.S. Girish sir, who is the chairman and professor of the Department of Biochemistry, Tumkur University, and also a good friend of my Professor Anand from Gulbarga University. So, sir called me, Professor Kumar sir called me to give a lecture. So I was thinking that what to give. So specifically, since I am addressing postgraduate students, I thought keep less of research kind of a work and then more of a learning based kind of a thing. So in that context, I'm going to tell you about the understanding of learning and memory with the subtitle History and Science behind it. So exactly all of us know learning and memory is very important, right? So that's the reason I think we are all surviving. Without learning and memory, I don't think anybody would be happy, at least forget about surviving. So it goes back with the history. It's nice, right? Goddess of memory. If you would have had a goddess of memory which can give you complete memory to you, it is wonderful. You can go and pray to the God and get the memory whatever you wish. Correct? So this is a Greek history which speaks about 3000 to 11,000 BCE. It's almost a Bronze Age, which was a civilization where the bronze tools were used. At that point of time, though, people realized that there is a goddess which is called a Mnemosyne. Mnemosyne is a goddess which gives a memory to the 9 daughters. These 9 daughters, which are having a different kind of a characteristics like a epic poetry, history, mime, flute, dance, choral poetry, and tragedy, comedy, and astronomy. Probably the civilization was thinking that to explore these very much, these topics, so which deals with always a memory, which are connecting with the discipline of this. So that's why 9 uses. Daughters were born, and all these daughters were giving a memory. Imagine like it went on and centuries together with the same kind of a concept. Of late, people have realized that there is a mediator. We do have a priest which brings a bridge between a God and us. In the same way, here also in the Greek history, it talks about Troponius, is the oracle of Troponius. So this person particularly is being called as a Greek hero or a demon who was swallowed by the earth and became the oracular god. So he was being prayed for a long time. So oracle is a person or a thing considered to provide an insight and a wise counsel and prophetic predictions. I think we have heard about oracle company, right? The Greek word has been taken and then we had company, right? Which talks about providing an insight and a wise counsel and prophetic predictions. Of late, this god was been believed to settle somewhere here in the lakes, in the caves. since it has been eaten by Earth, Mother Earth. So that's how the lake, there are several lakes in even Italy, Greece, and many other part of different countries, European countries. They had lakes which can give rise to a memory. If suppose some person has been been having some kind of a madness, who is not having a proper memory, all such people who used to go and visit this river and drink the water from there, and the disease gets cured. This was a strong belief. I think even recently, if I'm not wrong, where Biligiri Ranganabetta is a very well known, with such kind of psychiatric patients have been taken to a god and then made so many kind of pujas and here and there, even in the recent years, have been followed as it has been developed. And the Greek civilization, at the same time, Indus civilization also is happening. We are nowhere behind of any other kind of a civilization. Even we do have some kind of a representation which gives us a memory, right? Lord Ganesha gives a wiseness, and Lord Saraswati gives a knowledge. These are the two representatives which we do have about a memory, right? So we have not even left the Ganesha, each and every part of Ganesha, and has been given with all, see, big head with a big intelligence, focus and concentrate and desires and so on. Whatever we wanted, we have described very neatly that Ganesha gives the knowledge and wiseness too. It is a strong belief. So let's say now what is this learning and memory then? As we know now, and not only the current one, let's say even historians have noted one of the a statesman and a philosopher during 106 and 43 BCE. So he says, sweet is the memory of the past troubles. And the other person, Dante, is an Italian poet and a philosopher. There is no greater sorrow than to recall a happiness in times of misery. And another person called Enrico Fermi, a physicist in 20th century, he said he's called as the father of a nuclear age. He says, if I could remember the names of all these particles, I would have been a botanist. Probably botany people would appreciate how many plants and how many diversity, taxonomy and so on, you need to have a memory. So he compares with that diversity with the particles he observed in his discoveries. And even Plato calls the memory is the preservation of a sensation. I think this guy is quite sensible, right? He says in a preservation of a sensation. And the Aristotle was a Plato student. He says, colleague at the time in Athens during this period. So they were able to understand that, preservation or perseverance is called as a perseverance of a sensation. Perseveration of a sensation is called as a memory, right? So now we know that what exactly the learning and memory. So just let me define what is learning. Learning is nothing but a neuronal mechanism by which individual changes a behavior on the basis of a past experience. So hopefully you understand this concept of learning and memory before the class and after the class. That is the past experience which you were not aware, and let's say after having this event you would have a different kind of a memory now. Learning. And then the memory. Memory, you would definitely go and tell to somebody that, okay, these are the things which you have learned from this class, let's say. So that is an acquisition. First you store it in your head and then retrieve it with the sensory information. This is how it happens. Let's say there are different kinds of learning as such which have been classically explained. That is the reflex learning, which is associated with the immediate behavior change. And there is another called incidental learning. The behavior change, not immediate, but Person gets information from sensory inputs to develop a potential, behave differently. This is the kind of learning. Let's say in that reflex learning again here. You have two kinds of reflex learning. One is a non-associative, another is an associative. What is this non-associative? Is something like this. You are having a house somewhere in a peacefully, some a good environment. Suddenly you shift it over to the railway station nearby. So you will not get a sleep, right? Suddenly it happens. This is a kind of a severe disturbance, or else even in the market. But sometimes what will happen, same kind of a stimulus, the person learns about the properties of a single stimulus when repeatedly exposed with the same stimulus. For example, let's say train comes and goes, train comes and once it comes you get irritated, and several times it comes and then slowly get adopted to that situation, and then you will get adjusted to it. So you don't have further sensation now. So that is a kind of a non-associative that can have get an habituation or some kind of sensitization too. You can get even sensitization. You can get madness out of that also. So anything can happen with that kind of a non-associative. An associative, this is a classical example, probably all the science graduate would be aware of it, right? A person learns about a relationship between a two stimuli and a stimulus and a behavior. Your behavior will change. As soon as the school bell rings, what do? You take the bag and go away. Correct? This is what you used to do in school days. Correct, no? Let me tell you a joke. All mad people were taken into a flight accidents. That one mad fellow, he went and rang the bell. ಏನಾಯ್ತು ಅವಾಗ ಎಲ್ಲರ ಡೋರ್ ತೆಗೆದು ಎಲ್ಲ ಆಚೆ ಬಿದ್ದು ಅಂತೆ ಡಡ ಅಂತ ಫ್ಲೈಟಲ್ಲಿ ಎಲ್ಲ ಫ್ಲೈಟಿಂದ ಬಾಗಿಲು ತೆಗೆಯೋದು ಸ್ಕೂಲ್ ಬಾಗಿಲು ತೆಗೆದು ಆಚೆ ಓಡಾಕ್ಬಿಟ್ರಲ್ಲ ಫ್ಲೈಟ್ ಎಲ್ಲ ಖಾಲಿ ಇವರೆಲ್ಲ ಕೆಳಗಡೆ. So just it is an association with the ringing a bell and then you will have something like that. Otherwise you will not get thirsty also. Hotte kuda aswala. Bell haktidange hotte aswalte. So something like this. Right? This is the kind of a classical example people have demonstrated too. So coming to the another incidental learning is that incidental learning includes someone who learns new words while reading a book. You would have come across many words, buzzwords, right? Bioinformatics. You would have not heard about it earlier, right? Oncology. Let me tell you one more joke. In Tumkur Science Center, I was a graduate and I was working something like a part-timer. Voluntary basis science center. No, I was an active participant in the science center. We had one big program to say that a solar eclipse. There are misconceptions, right, on the solar eclipse. We should not eat at that time. We should do puja. We should do that, this, and all so many things, right? And pregnant women should not come. All sort of things, right, which we do not know. So that's why the science center had a big gathering, make everyone to come and see that through the telescope what is that celestial event which is happening there. On that time, there was books were organized in the evening hours. And that event was observed by one of the Swamiji also. I was in that book counter. He came and saw. There was a book called Oncology. All of you might have heard about it, oncology. What is oncology? Very good. Last bench, I need some sound. Oncology, all of you know, right? There was the one Swamiji came and told me, oh my God, this is the one. He knows how to read English. Oncology. That means our ancestors know very well about a cancer very long back. It is a oncology. It's called as oncology. So you can perceive however you wish. It's up to you. This is the kind of thing, like incidental learning. You come across a new buzzword, you will pick it up and then you will adopt it. So that is the kind of a thing. Or else, someone who learns the social skills while playing with others. So many times you would not know how to play the card. right? You go get out to the gathering, learn from them, and then play. So that's it. Many games, no, indoor games and all, you would not know, right? So such kind of a thing. Or else sometimes historical facts you will learn from watching a TV or listening to radio or something like that. These are kind of an incidental learning, right? These are the kind of learning things will happen. Coming to the, let's say, a memory, if you look at it in the same way, we can address it as a declarative memory and a procedural kind of a memories you have, different kinds of memories as such. So usually the declarative memories which goes with the facts and events, which you have learned the facts, geography you have learned, correct no? So anyone, can you tell me what is this one, this continent? Very good. See, your memory is working. Fine. I was thinking that it is India. Right? Certainly, yes. I ask you, like, what is the capital city of India? Very good. I thought Washington DC. Correct, no? No. You are sure it is the Delhi, not Washington DC. These are the facts which you have learned already. It will not change now until unless that order comes and Bangalore becomes the capital of city, capital of India. I do not know. So this is the facts which you learn. It is a declarative, very authentic one. So there is another kind of memory, events, based on the events. You go to naming ceremony, you go to marriage and all, you always link, oh, this uncle got married to that auntie. Right now? Sometimes you see somewhere, on the road, this auntie is a different no. Right? You will all have all suspicious, right? A declaration, you already declared it. Yes, such kind of a declarative memory, and then you will come to know that no, that auntie only your mother will tell, and then she has been put up so much of weight. Marriage was like that, and this one, and then you will realize, oh, she has been changed, but she is only. Maybe such kind of a conclusions you come, okay, after discussion. So these are all kind of a declarative memory as such you will have, and some kind of what you call the non-declarative memory. So the non-declarative memory is something like skills or habits, maybe a cycling or a music. Immediately you will not be able to learn. Over the period of time you will learn, and then once you have learned so fluently, easily, right? Flawlessly you will sing. I will not be able to sing, correct? The person who has not been trained will not be able to sing, and a person who has been trained, he can or she can sing very nicely. This is the kind of a procedural memory which will happen usually. And then another classical conditioning which makes you a different kind of a learning, as I said. So associative memory, you remember, right? If you give a bread, then the dog learns. At that point of time, okay, it will secrete its saliva and other things. ನೀವು ಏನಾರ ಅಂಗಡಿಗಳಿಗೆ ಹೋದ್ರೆ ಮೈಸೂರು ಪಾಕ್ ಅಷ್ಟೇ ಇದ್ರೆ ಅಂದ್ಕೋತೀರಿ ಅಲ್ವಾ? Am I right? If you like Mysore Pak, otherwise not. So another thing, like snake. If you see a snake, what you will do? You will take nice snake. Let me kiss the snake. You do that, right? No, you will get right. Suddenly you will be no more in that place. So that's the kind of a thing. Emotional responses. So these are the kind of responses which makes you to learn certain things, right? So as we all know now very well, since a basic science student would all aware now, right? The major learning side is the brain and the other one. So put together, spinal cord and brain together, it's called a central nervous system, and the peripheral nervous system which gives you sensation, right? So this is the kind of thing, very very important, brain. All of you would have hold this brain, right? How many of you are able to see and hold the brain? Human brain. Forget about brain, human brain. It's impossible, right? You can't even touch. We had that privilege of touching the brain, human brain. So at demands. So there is a brain cutting session and also brain bank is there. I'm not advocating, canvassing the demand, but you must visit the wonderful organ in the body. The brain is a part of central nervous system that controls the cranial cavity which we present. This plays a very, very, very important role in a memory formation, I should say, as a whole. So Carl Spencer, this is a doctor and become a scientist. So many doctors here, they go always a practicing, never do a research in their life in Indian scenario. But Westerns, all doctors, having spending like 10, 15 years of their time for getting a doctor, and they get into a PhD of 5 more years and spend almost their life, half of their life. And maybe at the age of 45, 50, they might get a MD and PhD at US. So that is a craziness of a Westerns are looking at the science. He is a psychologist and a behaviorist. He is the one who started doing some kind of an experiment on rats and primates, monkeys. So primate research is banned totally in India, right? Lot of ethical problem, Even using a rat itself is very difficult. People will come, right? They will do all straight and they will make it to stop. Okay. So, if you would have conducted in India the same kind of experiment, even at this point of time, you would have been behind the bar. That's the scenario, right? So he was working on this rats and monkeys, and then he started picking it out and then doing the surgery because he's a physiologist as well. So he could able to dissect it out and then do some kind of a removal of a part and then bring about the switches and then get back the animal and then try to do some kind of an experiment. This is how we started exploring it. And he could able to see that there are some kind of a different parts which do play in our learning and memory. Right? So today we know that so many parts of a brain, different parts of a brain. It's not a big deal. Even in a high school or a college book, you do see this, different parts of a brain. Majorly like forebrain, midbrain, and a hindbrain, which do have different parts as such. So you know that, okay, the list. See, this is the premotor area, frontal lobe being present, premotor, motor, and Broca's area. And temporal lobe is having auditory, olfactory, and temporal lobe and Rinke's area too. And parietal lobe do have a gustatory and somatic area, and this occipital is a visual area. Like each one have the different kind of functions. Probably you see that if there is a damage in particular part of the brain, you will be not be able to do these functions, right? This was the kind of a contribution which he made when he was doing the experiments. See, functional areas you can see over here and different views as such in the brain. This is a lateral view, this is a sagittal view and a dissected one, and then this is a superior view, and then this is a inferior view. You see that each part has been labeled over here. Visual area, association and a motor function area, Broca's area, auditory, emotional area, sensory, olfactory, sensory area. So different parts of brain do play very, very important role in a different functions as such. So what you learn now by looking at this picture, let's say, by looking at this picture itself, what do you think now? Safeguard your brain, very, very important. Put a helmet when you are riding a bike. If you learn that much is more than enough for my lecture. This is a Paul Broca. He found out a place, what is a behavior of a patient called as a Broca's aphasia. It is a deficit in ability to produce a language. Comprehension is generally preserved. I know I'm not to express. I know it. You need that condition? Nobody. Nobody willing. Right? I know I will not be able to tell. If I put a videos of that kind of a patient, he will be really mesmerized, right? That is the kind of a area which and that is the reason the area is labeled in his name. Is the Broca's area, small area is here. So here now, still now they say that the exact function of a language production is still unclear. Maybe involved with the muscle movement of a speech. Muscle movement, if it is not there, the tongue will not move. If I do not do these things, I will not be able to speak. Right? Whatever I am speaking, my vocal is working, and because of my muscles are quite active and which are being coordinated with what I am thinking and I am talking. So this is a very, very important muscle movement of a speech, and a syntax, grammar, verbal working memory, all of the above together as a bunch comes as a voice and try to communicate with you. Otherwise it doesn't come as such. Also thought to have a non-linguistic functions too. So this is a, as I was telling you, there is a Broca's area, there is a Rinkus area, there is another scientist called Rinkus, and he found out another area. See, let me tell you here, if there is an impairment of a speech, ability to understand and language is retained. I know, I'm able to understand what I'm telling, I'm not able to tell anything. Patient will have such situations. One patient who have a problem with the Broca's area. That is the motor function associated with the speech production. Then Rinkus area, responsible for the understanding of a language, vocalization retained, what is spoken is a total nonsense. I understand too, I know what I'm speaking, but when I'm producing it, you sense it as very wrong. My lecture will be very good no? If I speak like this, something like this. Is it fine for you? No, not at all. So this is the situation a patient comes into if there is a damage in the Rinkus area. So he will be able to tell the motor functions are doing good, and then he can move the tongue and everything. Because of the damage, he will not be able to put the tongue into a right place to produce a proper vocal. So patients do have such kind of situations too. I mean, this can happen either by a stroke or some accidental or sometimes an infection. Anything can bring about a damage in this part of thing, and people who end up with such kind of a lifetime problem. There is not much about a cure as such. Let's say, having done that, so you understand now the importance of the brain, and you must safeguard, and it plays a different roles. Then where is the site of memory? And don't advocate that, okay, sir has already told there is a site for a memory, only that site will keep and rest of the site let it get damaged. No, never do that. I will tell you. Right? Site of learning and memory. Certain structures like the cerebellum, hippocampus, amygdala, known to play a key role in the process of learning and memory. These are very important portions of the brain that plays a very important role in a memory. Most available evidence suggests that functions of memory are carried out by the hippocampus and other related structures. See, this is the structure of a hippocampus. I mean, here, the brain part inside. If you take sagittal system, cross section of the brain, you would see a part of a tissue in the same fashion of hippocampus. Like a seahorse. All of you might be knowing in a different, it's a seahorse. There are 46 species, it seems. Just I copied, in Google and then find it out. 46 species of small marine fish, the genus that are hippocampus. So, since the portion of the brain in the sagittal area, so inside, this is the one part. Suppose if you remove this part, learning and memory is gone. Is something like your head of the department. If your head of the department will not be there, then you see the department, how it will be. It will be very beautiful, you will be organized like this, and then the lecture will be going on, right? Is it true? Never happens. Never happens, I tell you. Nobody can take a lead and organizing such a wonderful lecture too. So this is what is important. In the same way, here this part is very important. It doesn't mean that he is alone doing it. Hippocampus alone will not do, but it will coordinate with a different part like cerebellum, and amygdala and so on. Let me tell you a little more about amygdala a little later. Let's say when we say that memory, you have a kind of two kinds of a memory, explicit and implicit. Explicit memory, if you can consciously work to remember. In the bus stand or somewhere here and there and all, you can do the mathematics, right? You can do, right? Somewhere on the road, bus, here and there, you can do maths, integration, differentiation. In Kundre Madakala, sir, in Basal Madakala, Adike sir, now we have left long back mathematics and all, right? Maths is allergy, that's why I become biologist. Correct? No? I know that. So, mathematics, solving a problem are very, very important and crucial. You put wholesale yourself, complete effort you will put it to understand and solve the problem. That is what we call it as an explicit memory and an implicit memory. In the bus and all, you will put some music and then you will keep singing. Somebody will say, "Hey, see, somebody is seeing you. Don't sing, loudly." Correct? No. You will, without forget, you will sing loudly. Correct? No. So these things happen. Easily it will happen. Implicit memory. So whatever the kind of memory, it goes like that. The memory as such, how it's stored and retrieved whenever you want, is very important. It's very, very important. Usually, when you have joined here in the university, you have a new friend. And she takes you to her house, somewhere in the street, down, and somewhere in that corner, on some small gully, and then there's a small house there. Right? The next time you feel like going it, and you will go, but you will not be able to find a house. Correct, no? It happens, right? This is what we call it as a learning and memory experiment. It is called a cognitive experiments. So you see here, this one, big street, particularly metropolitan city, and then finding some small house over here is quite difficult. So this is what effort is being done. Like what you do is now you picturize when you are going to your friend's house. No, you will know. I'll have to go by bus 5 kilometers, and there is a small village or something like this, a small town. On the way, there is one river comes. Soon after that river, there is one paddy field comes. Then coconut tree comes. Then there is a small village comes, and then there is a corner, there is a bakery. So here I should get down the bus. Like this you will memorize. Correct, no? All of you might have done this work. Correct, no? What you do is that you will picturize as the images. Those images will be kept in your brain, and then it will be recalled as an image here and there, whatever you feel like comfortable. And there is a blue gate in front of the house. If somebody also blue gate, another also blue gate, then you will get confused. That happens when you go to some kind of quarters, no? You will look very blank. Dumb, you will become totally dumb at that point of time. You will not be able to find out. But that's how you remember it. So that's the reason scientists also have adopted such kind of what you call as experimentations to conduct experiments in an animal. Very simple experiment. You have something like this, a simple what you call as a platform, right? So water maze experiments. So a trough which contain a water, let's say. So you allow this animal to swim. It doesn't swim. It swims, but it escapes from swimming. You also, if you are put into water, what you will do? You will enjoy and stay there only? No. You will try to come out of it. Right? You will look for the platforms here and there. Some platform is there, then you will happily come and sit. Correct? No? This is what it happens. So even animal also will be doing the thing. You put on platform over here. So let's say this platform it will learn within no time. You just put an animal in a water which is having a platform. It will do like it will go and sit there. whatever the kind of thing, and it will learn, and the shortest possible route also it will learn. It will go in the beginning, it will in search of a platform, and then later slowly goes and then sits there, and then learns. And then another time you leave, and then now shortest possible route it will find, and it will go and sit on the platform. This is a learning now. Now what I do is that, see, Indian scientists, I tell you, are very appreciable, I must say. You put casein over here. What will happen? If you put a casein into water, it becomes opaque. Wait, platform will not be seen. Now it gets confused. So you will make such kind of an experiment. ನಮ್ಮ ಹುಡುಗರೆಲ್ಲ ಏನು ಮಾಡ್ತಿದ್ರು, ಒಂದು 2-3 ಲೀಟ್ರು ನಂದಿನಿ ಹಾಲು ತಂದಿಟ್ಕೊಂಡ್ರು. ಅದಕ್ಕೆಲ್ಲ ಹೇಳಿದ ಇಂಡಿಯನ್ಸ್ ವಿ ಮಸ್ಟ್ ಅಪ್ರಿಷಿಯೇಟ್ ಅಂತ. We don't go with casein. It is very costly. So 1-2 ಲೀಟರ್ ಹಾಲು ತಂದ್ರೆ ಅದರಲ್ಲಿ casein ಇರುತ್ತೆ, ಹಾಕ್ಬಿಡೋದು. So that's how we used to do some kind of an experiment. I mean, my friends used to. I never did this water-based experiment, let's say. But this is how learning and experiment do happen. All of you are aware now. What is happening in the brain is that the process of learning begins with the sensory of a nerve cortex. As I said, cortex process and transcribe and sent to hippocampus and memory being formed. See, this is what happens. So the hippocampus and the cortex and the amygdala and different part of the brain have been coordinated. Visual cortex, if it is not working, then you will not be able to see. You go to the ophthalmologist, he says, your eyes are perfect, man, no problem at all. But the problem, then you go to neurologist and see. And why don't you get it, some MRI and done and come. Your eyes are perfect, but you have lost your vision. Because visual cortex is not working. Same way you can have an auditory system, hearing, right? So something like this. It coordinates. That's the reason I was telling you, safeguard your brain, entire part of brain. Don't think that something can be damaged easily. So it coordinates. I see and I learn, and I hear, I learn, right? I do touch this and I learn. It is a hot. I should not touch. Maybe a small kid, they will touch the file. With your experience, you say, don't touch. Then it will learn. No, it is dangerous. Something like this. It is a coordination process. So if the signals are strong and repeated, then what happens is called as a long-term memory being formed by sending a message back to the cortex. If the lesions are formed in the hippocampus, leads to the impairment in the memory formation. If there is a small damage in the hippocampus, then learning goes off. So as we all know now, the learning goes with the sensory nerves in the cortex. What these neurons, nerves do is that they communicate each other, one neuron to the another, through a chemical called as a neurotransmitters, right? And sometimes it can reach out to what you call an electrical excitability too. So the process of connecting the two neurons is being called as, it can be a dendrite, axon dendrite, or dendrite to directly a cell body too. So synapses being formed, right? And then leads to what is so called as a process called as synaptic plasticity, which I will tell you a little later. Let's say synaptic plasticity. Yes, we are only in this slide. Synaptic plasticity. The synapse is being formed. You know that cells are being formed once, and they will not again and again born, and then new cells will not born once you have grown up. Only thing is that neurons need to get connected. They need to establish a connection. Then only the learning to happen. Such kind of an experiment being done in the C. elegans. So this is an organism, a small organism, which is being used to demonstrate at a cellular level what is happening around. So you know how many cells would be in the C. elegans? Only 302 neurons are there. Only 302 countable neurons. Forget about our brain. Okay, you have billions of neurons there. So such complicated nervous system to read, understand is quite difficult at a cellular level. Certainly, as C. elegans being used to understand. So, if you give a stimuli, the reception which leads to a learning memory, that physiologically being noted as a plasticity. It can extend. it can extend, make connection with another neuron. This is being observed very clearly. So you need to do such kind of an extension as much as possible. Who are the winners in the recent word is that the people who are having the greatest network. Correct? No? That's why you are all on the Facebook, on LinkedIn, here and there, right? You will have more network. I don't know whether useful or not, but still you will have. Just to follow the reels. Right now? But no, you must have a network. No doubt about it. You must have a good network. That is very, very important. So that's what happens in good networking, makes you to learn more and more. Let's say that can lead to a new neurons and new synapses. New neurons usually, very rare it happens, from stem cells can arise. Very rarely it happens. But new synapses, which makes you to learn better, let's say, leading to the neuronal plasticity. So let's say now, what is happening here at the molecular level, at a cellular level, let's say probably now you are all interested to know, biochemists appreciate from here onwards. So far I told you about a history of development and anatomy and physiology. Now you are a biochemist. You always say like, I am a biochemist. I talk about chemistry part of it and chemicals and so on. So what exactly the signaling which happens, the neurotransmitters, which will presynaptic to postsynaptic, right? It do happen, like vesicles, neurotransmitter will be released and there will be signals. And when the neuron communicates with another neuron through the synaptic vessels, is that through an action potential. There is a membrane potential increases. You can implant an electrode and find it out there is an electrode potential to happen. Electrode potential should happen in such a way that it should bring it to the action. You never do that. One goal turn whatever correct, no? That leads to an action potential. Many potentials or many stimulus may not end up into an action potential. They may go up till here and then just come down like this, and it will not get to the membrane potentiation, will not bring it to an action. I mean, the next neuron should get stimulated. So that's how it goes with. Let's say this is the kind of a thing which will make that neuron to get excited and then make the another neuron to get excited further. So may not lead to every neuron with every same kind of intensity with an action potential itself. It may go slowly diminish also, depending on the kind of neurons. And these kind of stimulus, we call it as what? A potentiation. Potentiation. It is being observed. A long-term potentiation is a persistent strengthening of synaptic connections induced by a brief period of a high-frequency presynaptic activity. Many central synapse displays LTP through the required patterns of stimulus and induction in it, depending on the synapse. So the long-term potentiation. Now you see that in a beautiful biochemistry. You enjoy this, right? Correct, no? This is what you really enjoy and write everything. One neuron is communicating with another neuron through all these molecules. You can see that there are many cellular signaling to happen, and finally bringing in what? Neurotransmitter. See, for example, glutamate. Glutamate is being released over here, and then coming over here, glutamate receptors will receive AMPA receptor, NMDA receptors, and so on, and then make these molecules to get excited, protein kinase to ERK, and so on, a cellular signaling pathway, and calcium, all these things, calcium release and calcium mediated signaling to happen, see, nitric oxide, and so on, and leads to phosphorylation, dephosphorylation events, and finally bringing some kind of what you call as an action potential in the next neuron. So this will make the neuron to learn further or to keep those, nobody knows that what kind of a molecules really help to keep and bringing in a plasticity and then keep the memory as such. But nevertheless, you can narrow down to that. The CREB particularly, the CREB protein, which enters into after phosphorylation, let's say, they stop and then express some kind of what you call as a genes as such. This has been very well addressed, and particularly the kinases. Kinases are being documented very well documented for bringing about what you call as a long-term potentiation. Long-term potentiation. Long-term potentiation is very important for learning. This was a school of thought over the period of decades, several decades, I should say. So long-term potentiation is important. But of late, there is another school of thought. A scientist they think that no, not only a potentiation alone, long-term depression too is important. So that's where people have come up with a cerebellar long-term depression is a type of synaptic plasticity has been considered as a critical cellular mechanism for a motor learning. LTD occurs with an excitation of a synapse between the parallel fibers, Purkinje cells, cerebral cortex, and expressed reduced responsive in the transmitter to the glutamate. So now there is a potentiation and there is an active depression, let's say. Both are important. Both are important, I should say. So then only the learning do happen properly, right? So the long-term depression, which goes with what you call the NMDA receptor with the calcium mediated, and then what is called the calmodulin comes into picture. Whenever there is a calcium being increased, a calmodulin do get activated and brings about a kinases 2. At the same time, another enzyme called the calcineurin, which brings about what you call the LTD and LTP. The equilibrium you can see over here, LTD and LTP, which is going with the synaptic protein getting phosphorylated and dephosphorylated. Let's say, in nutshell, the memory formation happens from here. See so much. Okay, I'll not go in detail. So this is an MIT news in recently 2018, if I'm right. Yes, 2018. From the website only I have taken this image. This image shows the neuron CA3 region, a particular hippocampus region, which is important in memory encoding and retrieval. Nuclei of CA3 neurons are labeled with the green and the dendrites are red. A smaller spike green above represents the axon projecting to the CA3 regions of a dendrite layers. As I was telling you, the synaptic plasticity has been established even recently, 2018 article. So the protein kinase, I was telling you, right? Protein kinases are very, very important. Protein kinases are very, very important for the long-term potentiation. At the same time, as I said, long-term depression is important. This is the person, a few experiments he made with learning and memory. But this guy is called Susumoto Nagawa. I was indeed very happy to meet him at the NCBS once when I was a research scholar. So I could be able to hear his Nobel lecture at NCBS. So this guy, he gets a Nobel Prize for Physiology in Medicine 1987 for Susumoto Nagawa, the discovery of a genetic principle of generation of an antibody diversity. He is a molecular biologist. But he was the one who could address this with his new techniques or technology to see that there is a molecule which is very, very important in learning memory. So he could be able to establish, and this is the one. Evidences associated with the schizophrenic genetic variation with so and so PPC. So you see, this is being called as a enzyme and conditional calcineurin knockout. He could be able to knock out this, and particularly with a horrible experiment, he could prove that the learning and memory will not happen without this molecule. So let's say, what is that molecule is all about? A calcineurin. The calcineurin, you can see that the long-term potentiation, long-term depression do happen between the neurons, and this is simply a protein phosphatase which regulates the second messenger and a calcium in the central nervous system. They play a very important role in the learning and memory. And the multiple functions of what you call as the calcineurin. You know why this calcineurin, being called as a calcineurin, is because it is a calcium dependent enzyme and it is rich in the nervous system. People have, after that incident, people have done all kind of an experiment with the radioactive labeled protein and radioactivity experiments and see that the entire brain is loaded with this molecule, let's say, right? So without this, let's say you would develop such kind of a problem, a psychiatric problem, you will not be able to learn in your lifetime. You will not be able to learn anything or keep a memory as such. So this is the kind of an enzyme, very simple enzyme. As you know now, enzymology you might have learned so much, right? It is a simple enzyme. One is a catalytic active domain. This is a regulatory domain, and calmodulin with the calcium, and calcium also binds with the calmodulin. They all together bind, and this is a kind of what you call as an active protein which can dephosphorylate the substrate. This is the substrate. What it does is the simple dephosphorylation process. That's it. Removal of a phosphate. Very good. Last bench in the Bandhan advisor. Thank you very much. You reduce my effort. Right? So catalytic domain and so on. Let's say this was the story which we began. Let me stop here or continue little more.
Please continue.
Continue.
Yes, sir.
This was the work which have been assigned to me as a research scholar by my boss. He made me to exact with by telling all this kind of a stories. Let's say maybe I put my own slides there, but he told me like calcineurin, there is an enzyme which does this, that, and all. Why don't you start working here? Come on, man. We will go to the library and go and find out some articles like that. He took me, and then you know what happens after doing the research, right? So this is the thing which happened. Let's say what was an idea is that calcium dependent particularly calcineurin to assay in a WBC, platelet, and a CSF. This was an idea. But my boss idea was that why don't we able to estimate the phosphatase to be in a CSF samples of a neurological disorder which comes to a nuance. That was an idea. Sometimes let's say it's a simple, as somebody said, it dephosphorylates. What is the substrate? Can anybody tell me what is the substrate you use for a phosphatase? What happened? Yes, sorry. This is the substrate, para-nitrophenol phosphate. As you know, that calcium dependent enzyme, you put a calcium into the assay system, phosphatase must work, put the brain sample and put a PNPP, it should work beautifully. So simple, no? Put whatever the buffer and all those things and then put calcium and then see the activity. You will get wonderful, right? So having started digging out several kind of papers, and then I could find out that it never works on the calcium, rather it works on a manganese, cobalt, nickel and so on. Where to find a cobalt and a nickel in the system, physiological system? No. But when it comes into the test tube, it works better with a nickel and cobalt. People have realized. So they started using a nickel and copper heavily, and then started doing a diagnosis, and then people have done with all sort of work with that same kind of a methodology as such. My boss was not willing. There are not many protein phosphatases too. You see that list. And this is the enzyme, a protein phosphatase. It can be inhibited by certain inhibitors like iminophilin cyclosporin, and people used to use some kind of inhibitors and find out the total activity and then minus it and then see that there is the activity for the calcineurin acid. This is what I should say, cumbersome procedure to assay this enzyme. Right? Why para-nitrophenol phosphate we need to use? This is unnatural. That was my past judgmental. And a manganese. Two are unnatural totally. Why do you want to do this? We do use natural one, at least near to the physiological condition. And a phosphotyrosine being used, phosphopeptide being used, and a phosphoprotein being used for the assay system. Probably I struggled with the spectrophotometer for a long time. Right? This is what exactly, and we have done all that regularly do Lorentz method protein estimation, right? That way we can estimate a tyrosine, right? So that's how we have tried all those circuits I have done, but maybe years, almost 6 months to a year, we were not able to find out the exact, because the proteases also do increase this, and protease inhibitors you need to put in now to the system, so that block the proteases, measure only protein phosphatase activity. As you know, now there are several protein phosphatases, you need to block everyone now. So quite cumbersome procedure, it becomes very costly, right? So that's the reason what we thought, like we can use only phosphotyrosine like this, phosphotyrosine, and we have measured the difference between the phosphotyrosine and the tyrosine. There is a absorbance difference in there. When phosphorylation do happen on a tyrosine, there is a quench in the absorbance as such. You can see very easily. So this part we have done, and a phosphotyrosine you can incubate and measure at 280 nanometer and see that there is an increase in the activity as such. That's a beautiful story. Easy to say, theoretically. But practical difficulties do come. So nevertheless, I started working on this, and then we were able to establish with the phosphotyrosine alone, and we see that when we use a PNPP, as I was telling you, the manganese, cobalt, nickel and all, they are doing best compared to this, calcium. So our idea is to promote a calcium, because the calcium signaling, right, which is happening with the calcineurin. So we use the calcium with the phosphotyrosine and see there is an activity. You can see that the bar, this graph being cut, several folds has been increased with the phosphotyrosine when we have used. Whereas the PNPP very short, and even a manganese. That means we can measure very good activity. And as we know that in the system, just now you have realized, calmodulin dependent enzyme, I told you, right? Calcium, calmodulin. You bring the calmodulin also into the enzyme system. That's what we did. Calcium and calmodulin together. Calmodulin was purified again, another chapter for a thesis. Purify that and then get that calmodulin into the system. We have brought in, and then we got to see that there is an activity which was a fold increase with the calmodulin and without a calmodulin. You can see that without calmodulin is less, with the calmodulin has been increased. So that means we are measuring the calcineurin activity more naturally. That much I say. So this was a professor. I must dedicate this talk to Professor Tarana Shetty. He is no more now with us. He was a very good professor. He taught me more than a science, I should say, human values. I must thank him for introducing me a wonderful subject, neurobiology as such. So this is what our impression with the Professor Tarana Shetty used to come and tell me around 12 o'clock, 2 o'clock, or sometimes experimental work going on. He used to come and tap me saying that, you don't know the quality of work, what you are doing. He is a classical biochemist. You don't know, wonderful man, this is the kind of a work which will go with on par with the big paper like the Lodis et al. Your name will be known for everybody. This was the kind of pumping things which he used to do. But sometimes it happens unnoticed, efforts goes wrong. So we have put a lot of effort on developing that method, but we could publish into Indian Journal of Experimental Biology a novel assay method for a calcium calmodulin dependent phosphatase in the brain extract, bovine brain extract. So this is the low-risk method. You know that there was a survey which has happened in the, which paper would have been highly cited in the world. You know that? Anybody from the MSc student fraternity and research scholars now? Which paper has been cited highest in the world? There was a big survey in the world. What is the greatest science that has happened? Great science has happened. Is it a discovery of a DNA? Is it a structural determination of a DNA, the molecular biology? Is it cracking the central dogma of molecular biology? Cracking the genetic code? Finding the PCR? Which one? Any idea? You must be very happy. Day by day you do the same job. So this is the paper which has been highly cited. The most highly cited paper published in a protein determination. Every now and then you do protein estimation. Everybody cites this paper. This has been highly regarded in the scientific community. I think many JBs should have come, not to the quality of this work, right? So, though it has been published in a JBC, and a Nature people have published so many, but none of the contribution has been equated with the Lowry, and Lowry has been awarded with the highly cited publishing paper in the history of science. So he's the guy, right? Let's say, and all of you know that long-term memory goes with a different kind of intracellular calcium as such, a short-term memory and a long-term memory. These are the another two kinds of things which do happen, and particularly the phosphatases and kinases, dynamics do happen. So which brings about some changes in the what you call as the kinases and phosphatases, and with the intracellular calcium. If there is a calcium, you see here working, it goes like this. With the short-term memory, calcium goes like that, and the calcium increases this way, and phosphatases to kinases, kinases to phosphatases to happen. This phenomena must happen with the memory formation as such. So the another very important hurdle it came into my problem is a big problem, I should say, a shocking. When you are going through the review of literature, many times you do not do the review of literature properly. You just listen to your boss and say that okay, this can be done, and simply go keep doing the work about 1.5 years, 2 years, and you will come to know that already people have published this. That's why it is very essential to review thoroughly and find it out what exactly is happening. No, it happens with everybody, right? So one should not be ignored. But I was aware of it, but still, I'll tell you over here. This is the enzyme, calcineurin. The same enzyme is being present in the T cell as well. T cell. All of us know it plays a very important, another big system in the body. That is the immune system. Equivalent, more equivalent to the nervous system. Right? So the immune system comes into picture now. Let's say the same calcineurin does the same, increases the interleukins, these, and bringing about boost in the immune system. Boost is the secret of my energy, right? Huh? It's bringing a boost in the immune system. So is it an advantage or a disadvantage? It depends on the situation, sir. What, sir, you are telling? What a kind of question smartly you are asking? Correct, no? Yes, it is true. It depending on the situation. Boosting is not good. Correct, no? So particularly this is an understanding in the immune system has led another milestone of a transplantation, I should say. I will bring it to that level, let's say, the transplantation. So is that your immune system need to be checked. If you want to transplant a tissue, now system get accept slowly. So now people are very, very curious to see that block this enzyme, let's say. So people have published the concept of that, the activity of a calcineurin as such in the nature, and then coming up with what you call as the latest target this enzyme. Every now Tom, Dick, Harry have started targeting this enzyme at a different levels. Block this enzyme now. So that, see, BTP, trifluoperazine, autoinhibitor domain peptide, cyclosporine, FK506, UAT peptides, so on. So many companies have come up with a different level to block this so that it is under check now. Now organ transplantation is possible. The combination, cyclophilin, all of you might be heard about it. Cyclophilin. So if you inject the cyclophilin, your immune system comes down now. So slowly you can make that organ to get transplanted. Transplantation procedure has become excellent now in the medical world because of the discovery which has happened to block this enzyme alone, calcineurin. That's all. So now, let's say the activation and the inactivation. You block this by using this FK506. It has been called rapamycin, FKBP. There is another protein which goes combined and then makes a binary complex which blocks the activity of the calcineurin acid. So we also thought, by that time two publications and then you will have a PhD award and all those things will happen. I also got a PhD award, right? And then I also became a faculty in Bangalore University. It's a proud feeling. And I got a guideship also, the another feeling. I can also guide. One guy approached me, a poor fellow. Sir, I want to do a PhD under you. Oh, please come. Then, like my boss, how he made me to exec, I also told some stories about calcineurin. Calcineurin is like this, that, and all. What is interesting is that there is a part in the calcineurin that is auto-inhibitory domain. This is what I am interested in. There is auto-inhibitory domain. We will explore it. I will try to do something there, as if like we are doing some tailoring job. We can cut this and then do something. That's what exactly we did about 5 years with my student. So this is a kind of catalytic domain, this is the regulatory domain, this is the auto-inhibitor domain. You can see here from here to here auto-inhibitor domain, which is very important. You know what it does in the physiological situation? When you go to your head of the department, you will stand like this, so obediently. When you go to the field, you will play around. Correct? No? When you are with your friends are different. When you are with your home people, you are different. Like the calcineurin also behaves. It will hold like this auto-inhibitor domain until unless there is a calcium is coming in. When there is a calcium in the picture, now my game is started. So this is how calcineurin works. So here the auto-inhibitor domain, it works like that. So now what we did that we tried to dissect it out. So let's say now, as I was thinking, this is exactly the sequences people use. Sorry, this is the sequences. Auto-inhibitor domain, particular domain, AID domain. These are the amino acids being present in the auto-inhibitor domain. Can we do this auto-inhibitor domain? So this is what I saw. Okay, we wanted, this is what exactly the 3D structure of the protein, and we have modeled as such. Oh, sorry, let me tell you about it. So very importantly, as I was telling you, review of literature is important. I have realized over the period of time with my experience in the research, I told my student, you perfectly do the review of literature after giving a problem. Now slowly he started learning how to do review of literature. He came up with a paper saying that, okay, there are several papers which have been done somewhere 1990, 1995, 1996. 2007 I gave this problem to this guy. People have already explored. What they have done is that they have taken that auto-inhibitor domain, synthesized or whatever, and then they have done digestion of this auto-inhibitor domain by using a proteases, and then take out the small peptides and then observe the inhibited activity as such. So this is what exactly it has happened. So then my work is gone now, my idea. I cannot ask him to do PhD now. Correct? No? I should think other one. No, I told him, mission on a moon would have happened long back. But we are still hoping, right, in India, we'll take the man on the moon. So like this I thought, we'll take the man on the moon, no problem at all. We'll take the same problem, we'll use a modern technology to use. By then there was no molecular docking concept, there was nothing like that. So what I told him, like, you take the same peptide, as much as possible, small fragment, a possible smallest fragment, if it is going to inhibit the activity, this is your achievement. Fragment it and see that molecular docking through that, find out the binding affinity, and we will come up with some short peptides, a possible peptide which can really block this enzyme, calcineurin. That's what we have done it. And after doing that, we could able to find out P1, P2, P3 peptides, and there was already in the market, auto-inhibitor domain was sold in the market as such. You can take this auto-inhibitor domain peptide and use it in the assay, calcineurin assay you can do it. So we have compared that data with the auto-inhibitor domain. See that there is an activity normal, and by using this auto-inhibitor domain, this is the one. P1, P2, P3. There are 3 peptides which we have screened out of, let's say, almost 500 we have generated, 500 odd, okay, generated, all of them were docked, and then finally we have come up with the shortest fragment, and then with a good score we have synthesized, and then we have used in the assay system nearly about 5 to 6. Finally we have narrowed up to a 3 peptide. We could have patented it, we didn't do that, we have published it. After that nobody has interested to take this problem. So it is very difficult nowadays, giving an idea to the students and making them to work on our idea. They will come up in their own idea, cheap ideas, right? Sir, illiದೆ sir, ಅದನ್ನ ಮಾಡ್ಬೋಣ sir. ಅಲ್ಲಿದೆ sir, ಅದನ್ನ ಮಾಡ್ಬೋಣ sir. ಇಲ್ಲಿ ಇಡ್ಲಿ ಸಿಗುತ್ತೆ sir, ಇಡ್ಲಿ ಮಾಡ್ಬೋಣ sir. ಆಮೇಲೆ ಆ ಕಡೆ ಹೋಗಿ ದೋಸೆ ಮಾಡ್ಬೋಣ sir. Something like this, they will come in their own idea, which facility what they are having it, and this, that, and all they will do. No problem. It is our pet also, right? So this is the one, ask the student to publish a paper on that. And then another part is that phosphopentapeptide we have used for synthesize, and then we have used in the acid system. This is the another problem which we have solved. So having done my story with the history of learning and memory and history of my research work also. So just I will conclude saying that the neuroscience is a vast field. A technological innovations such as the brain imaging, artificial intelligence allow deeper exploration of learning mechanisms and memory storage. Applications like adaptive learning platforms and memory enhancing strategies based and transforming an education is a cross-sectional anatomy of the brain now. It is, why did I put this one? I have made a link also on this, but I do not know I will be able to see show that because internet connection need to be there and connection need to happen. Radiology has happened so that you can dissect out a brain without even physically touching it. So such a radiologist, radiology techniques have come up with a functional MRI to a functional MRI. So lot of data is coming up, and one can easily use and bring it to what is called as an artificial intelligence into the system and try to understand very well learning and memory. Very, very fine-tuned memory, how exactly it happens, what is the role of a particular, a pinpointed brain portion as such. So I thank everyone, and before that, just I wanted to give you, since you're all tired after listening to my lecture, all of you, right? Correct, no? All of you are tired, no? Yes. Honestly, you tell me. Don't lie. Don't lie. I know that you are tired. I am only tired, you are not tired. Just one. That's the reason I am giving all of you a juice. Okay? So that you get energized. Right? The juice is that all of you take a paper and pen.
Juice.
Paper and pen. All of you are students, right? Paper and pen you have? Okay. I read this list here. Okay? Try to remember and reproduce it. That's all I'm asking. Okay? I read now for you. 10 starts now. Spoon, bag, plate, scorpion, chair, pan, sweet, alarm, bent, chair, light, flower, curtain, wet, chair, door, dinner, plaster, tomato, car, papers. Right now. How many you remember? Let's check your memory now. I give a list, right? You remember, right? Write it. Let's see your brain is working or not. I don't give much time now. ನಮ್ಮ ಸ್ನೇಹಿತರು ಕೊಡೋದ್ರಿಂದ ಸ್ಪೀಕರ್ಸು ನಮಗೆ ವೆಹಿಕಲ್ ಬರೋ ಸಾಧ್ಯತೆ ಇದೆ. ಅವ್ರನ್ನ ಫೋನ್ ಮಾಡಿ ನೋಡಿ ಸರ್. ಇಲ್ಲ ಬರೋದಲ್ಲ. ಓಕೆ, ಐ ಥಿಂಕ್ ಲಾಸ್ಟ್. ಸೊ ಸಂಜೆವರೆಗೂ ಕೇಳ್ಬೇಕನ್ನ, ಪ್ರೊವೈಡೆಡ್ ಅವರು ಪರ್ಮಿಷನ್ ಕೊಟ್ಟರೆ. ಯು ಆರ್ ರಿಟನ್ ನೌ? ಆಲ್ ಆಫ್ ಯು ಡನ್ ನೌ? Very good. You must smartly admit ನಾನು ಎಲ್ಲ ಬರ್ದಿದ್ದೀನಿ ಅಂತ ಅಲ್ವಾ? ನೋ, ಡೋಂಟ್ ಡೂ ದ್ಯಾಟ್. ಎಲ್ಲ ಬರ್ದಿದ್ದೀನಿ ಅಂತ ಹೇಳ್ಬೇಡ್ರಿ. ನೋಡೋಣ. ಲೆಟ್ಸ್ ಸಿ ನೌ. How many of you remember spoon? Raise the hands. Spoon. Spoon. Quite a few of numbers are missing the spoon, right? No, nimmonne tarkari tanakku kalsutidane. Many of you remember this word because the primacy, which means we are good at remembering the first item in the list. ನೀವೆಲ್ಲ ಎಂತ ಮೆಮೊರಿ ಇದೆ ಅಂದ್ರೆ ಫಸ್ಟ್ ಯಾರು ಹೇಳ್ಕೊಟ್ಟಿತ್ತಲ್ಲ ಅದನ್ನ ಮಾತ್ರ ಜ್ಞಾಪಕ ಇಟ್ಕೊತೀರಿ, ಆಮೇಲೆಲ್ಲ ಮತ್ತೊಂದು. ಇನ್ನು ಕನ್ಫ್ಯೂಷನ್ ಬಂದಿಲ್ಲ, ಆಮೇಲೆ ನೋಡೋಣ. The another one. How many of you remember car? Car. Oh, you car. ತುಂಬ ಕಡಿಮೆ ಇದ್ದೀರಲ್ಲಪ್ಪ ಕಾರ್ ಬೇಡವೇ ಬೇಡ ಕಾರು. Okay, very good. So this is the resonancy. Many of you remember this word. This is because the resonancy is that which means we are likely to remember the most recent word heard. Car was the last word which I said. Last word, if you heard it, your auditory system is working fine. That's why you were able to remember it. The another one, chair. How many of you remember chair? Wonderful. ಅಂದ್ರೆ ನಿಮ್ಗೆಲ್ಲ ಎರಡೆರಡು ಸರಿ ಹೇಳ್ಬೇಕು. ಹುಡುಕೊಂಡ್ರೆ ಗೊತ್ತಾಗಲ್ಲ ನಾವು ಅಂತಿಲ್ಲ ಮನೇಲಿ. ಎಷ್ಟು ಹೇಳೋದು ನಿನಗೆ ಅಂತ ಅಲ್ವಾ? ಹೇಳಿದ್ರೆ ಗೊತ್ತಾಗೋದಿಲ್ವಾ? ಎಷ್ಟು ಸರಿ ಹೇಳತ್ತೀನಿ am I right? These are the categories. Right? Repetition. Many of you remember because it has been repeated 2 times, 3 times. Scorpion. Scorpion. ಇನ್ನಿದ್ದವೆಲ್ಲನೂ ಬೇರೆ, ಇದೊಂದು ಮಾತ್ರ ಸ್ಪೆಷಲ್. ಉಪೇಂದ್ರ ಅಂತಾರ ಡಿಫ್ರೆಂಟ್. Surprise element is there. You remember this word because you stood out of the rest of the list. Unusual word which makes it to remember and compare the bunch of the word generally being used. Who remember table? Table. Few of you remember table, right? I know that. It is a false memory. Sorry. It is a false memory. Some of you remember table, no? It is a false memory because you have linked up now. Chair, spoon, right? On a plate, whatever is there, no? All these things together, right? Chair, dinner, spoon, plate, they all come together and table must be there for that. So you keep one table also there. This is an extra addition you have made. Okay, this is a false memory. So like this kind of memories are there, right? So you can keep doing such kind of an exercise to improve your memory. I acknowledge all the group members. Let's say Karnatak University to Rona Ayurvedic Hospital and Tumkur University. Sir is here always with us. And then Defense Food Research Laboratory, having done from the HPLC there, NIMANS for advice and other things, and CFTRI for some part of work, and Chromed Bioscience where we are doing animal work particularly, and Viprogen for LCMS kind of a work which I've been doing now. So this is a little funding which I received from these grants. Without acknowledging these, very, very bad, I must say. UGC Government of India, DST Government of India, RGHS and UNIT Government of Karnataka. So these are the, without saying it goes, the effort of my students. He is a Dr. Gopi. He is the first student who works on a UGC program, and then he joined Thermo Life Sciences as a behavioral scientist, and later he started his own company in Bangalore. So he is doing excellent. So I think I want him to visit your university. Probably I will talk to sir because he has come up with a new product as a behavioral app, particularly for the students, for employability, how to increase the employability in the students' fraternity. This is the guy Harish, and she is Anushri, and he works in Bangalore University as a this faculty, and she is in Singapore, and she is working with stem cell biology work now. And these two were from Karnataka University, Arvind and Sarojini. Sir only, Girish sir only awarded a PhD for them, being an examiner. Both of them are there in Wisconsin University, USA. These are the people who are working currently. But as I said, they have come up with their own problems. I have asked them to do the work with my knowledge. See, it's very difficult to make them to work whatever we think of. So now we need to be a little bit flexible and ask them to do whatever they want. So that also I do. We have become almost saints, Some bless the children and said, This is what had happened. Teachers now, ಕಾವ್ಯ ಹಾಕೊಂಡು ಬರೋದು ಒಳ್ಳೆ ಸರ್ ನಾವು. ಇನ್ನು ಮುಂದೆ. Thank you very much for patience hearing.